Here we have part 2 of Lab 11, exercises 42 and 43. In this lecture, I'm going to cover the female reproductive system. We're going to start out with gross anatomy, and just like I did for the male reproductive system, we're going to start where our gametes are formed, kind of follow their path through the system. So remember, our gametes or sex cells are formed in our gonads. And for females, these gonads are called the ovaries. So ovaries are paired. There are 2 of them. And the ovaries are homologous to the testes. So that means that embryonically, they are derived from the same tissue types. So during development, formed from the very same tissue types that testes are. The big difference is that the gonads in females are inside the body cavity, where in males they were outside. And you can see here that all of the female reproductive organs are located within the peritoneal cavity, and they're all in retroperitoneal position, just like our kidneys, except for the ovaries. And we'll see that all of these structures are supported by a series of ligaments that I will cover at the very end. So the ovaries is where gamete development occurs. And in this case, we're dealing with ova, or eggs. So once our eggs are fully formed and ready to go, and we'll discuss this cycle that takes about 28 days, when ovulation actually occurs and the egg is released from the ovary, it is going to travel through the uterine tubes or fallopian tubes. Now, what's interesting is, unlike males, the gonads are not directly connected to this tube or duct system. So there's no actual contact between our ovary, our female gonad, and the duct system, which is the uterine tubes. Instead, we have these little finger-like projections called fimbriae that kind of cup or surround the proximal part of the ovaries. So the actual term fimbriae means fringe. So you can see it's a little fringe-like projection. And during the menstrual cycle, in response to hormones, these finger-like projections can become engorged with blood, and they kind of move in a sweep-like motion over that proximal end of the ovary. So that helps when the egg is released to kind of suck it up into the tube system. And the fimbriae itself, the inner surfaces, are lined with ciliated mucous membranes. But this is why some women actually can have ectopic pregnancies, or we can see inflammation in the reproductive system, because there is no direct connection between our gonads and our duct system. Okay, so our egg or ova leaves, the fimbriae helps sweep things along into our duct system, which is the uterine tube. And our uterine tubes are also paired, as you can see. And they are also lined internally. So the mucosal layer has ciliated columnar epithelium. So all the way along this tube system, we have little cilia that help move the egg along its journey towards our uterus. So the uterus is a kind of pear-shaped organ, where the egg ultimately travels to and would proceed to develop into an embryo if fertilized. And we're going to look at the inside of the uterus, what it looks like through a section, and microscopically in a few slides. We can see that the uterus, when we'll turn to the side, we'll see that it's where it's positioned and how it's supported. So this is the location that is going to house the embryo or the fetus the entire time during development. So as I've mentioned, this whole reproductive system is supported by a series of ligaments, and there are two that are pretty important. So we have our broad ligament, and that's just a fold of peritoneum that drapes over the uterus and the uterine tubes, so anchoring them in place to the body walls. And it's this yellow section on the torso model here. We also have an ovarian ligament that helps to anchor our ovaries to the uterus. So everything is held in place by a series of different ligaments and peritoneum. Now I'm going to open our torso model like a book so we can see all of these organs and structures from the inside view. So you can see the arrow. I'm going to open and we're going to take a look at the right anatomical side. And here we go. Let's review some of the organs we've already covered and structures. So the gonads are the ovary, not directly connected to the duct system, but kind of surrounded by those little fringes called the fimbriae. Connects to our uterine tubes that then move into the uterus. So here you can see that the uterus is positioned between the bladder and the rectum. And remember, that's supported by a series of ligaments. So the uterus is about the size and shape of a medium-sized pear. So it's about 7.5 centimeters long and 5 centimeters wide. And you can see that it's slightly flattened. It's very muscular and has a couple different regions. So you can see this kind of flattened round end that we refer to as the fundus. The majority of the uterus is the body, and then it has a narrow end called the cervix. So the uterus is where the fertilized egg is ultimately implanted, and it enlarges to house and support the embryo and fetus during development. Inside the uterus, we have a series of linings, and we're going to look at that in more detail on the next slide. But those cellular linings are what respond to changes in the hormone cycle. They swell and grow to help support and nourish that developing egg. And then ultimately, if no egg is fertilized, those layers are shed through the vagina. So our cervix, you can see, kind of represents the end or the cap of the uterus, and it's very narrow, and that's what connects to the vagina. Little side note here is that cancer of the cervix is very common in females, but fortunately it can be detected and treated. To do this, women are encouraged to go to the gynecologist annually to get what's called a pap smear. So when you go get a pap smear, the doctor is kind of scraping that cervix to collect these cells that line the edge of the cervix. And that's done so that they can take a look at those cells to see if any abnormalities are present. Okay, so the vagina extends for about 4 inches from that end of the cervix all the way down, and this is our main copulatory organ. So this receives the penis during intercourse, but it also provides a passageway for the delivery of an infant if that egg is fertilized, and also for the shedding of that endometrial lining of the uterus during the menstrual cycle. So the only external feature that I'm focusing on in this image here on our torso model are the labia minora. So labia minus is another term for this, that the tissue that kind of surrounds the folds or tissue that kind of surrounds the vagina that has lots of sebaceous glands. So produces mucus or fluid. Okay, so finally, we've got this little weird structure hanging out over here. This is the clitoris. We're going to talk about the clitoris a little bit more on the next slide. I want to take a minute and talk about the gross anatomy of the clitoris. So I've already pointed it out on the torso model on the previous slide. Here it's emphasized again. And this structure should look familiar to you. And that's because we look at the male torso model. This structure is homologous to the penis, right? So here's our homologous term again. So remember, this just means that during development, there are structures derived from the same tissue types. And this is the case for the clitoris and the penis. So this is what a clitoris looks like. We've got a diagram here, but you should notice some of the same features or structures labeled as we saw for the penis. So for example, we had our glans penis, which was the head portion. We also have a glans clitoris. So they look very similar to one another. Another thing that we see are the corpus cavernosa. Same thing in both of these organs. So remember when we looked at a cross section of the penis, the corpus cavernosa were these two tubes of erectile tissue that were paired down the dorsal portion of the penis. So what's interesting is if we take a look here at an image of the clitoris, we can actually see a section through it. So I'm just pointing out, here's that glans part, the head of the clitoris. You could see in the drawing, just not as defined in this image. But here we can see the corpus cavernosa. So similar to the penis, these tubes are erectile structures. So they expand when they are filled with blood. So it's interesting because these form or are formed from the same tissues, have the same functions. It's just that the clitoris is obviously much reduced in the female, and it's not used as a copulatory organ as it is in males. So these tubes of erectile tissue, just like we saw in the penis, are bound by the sheath called the tunica albuginea. Remember, that's in yellow on my penis slide here. So the only thing that we don't see, we obviously don't see a urethra passing through the tube because the urethra in the female is below the clitoris. They're not combined. So we don't have that corpus spongiosum. So the fact that these two structure and organs are homologous has led to some interesting evolution. Here's an image of a female hyena, and this is her clitoris. So it looks very much like a penis. And when we look at this structure, as it's all dissected out, you can see all of the components within the female reproductive system have actually merged so that it can form a continuous pathway from the uterus to the vagina all the way through the clitoris. So this means that the clitoris functions exactly like the male penis does, meaning the urethra passes through it. So they actually urinate through their clitoris, and, which is really crazy to me, they give birth through this structure. Not surprisingly, they often have stillborns, their first pregnancy, because this process can be quite difficult. So because these structures are homologous, evolution has actually shaped them to look very similar because the tissues are the same, a lot of the structures associated with them are the same. And it's not just hyenas, there are some other examples. So in certain species of lemurs, we see a clitoris that looks more penis-like, and in a group of mammals called the Fusa, which live in Africa. But hyenas are the most advanced kind of form of this evolutionary similarity. And one of the hypotheses behind this is that females actually are the alpha in hyena packs. Let's take a look at what the ovary looks like internally. So here I have a torso diagram. So you can see that when we take a section through the ovary, we can see a lot of different things going on. And when we look microscopically at this, we can see some of the different regions of the ovary. So we've got this end, which is the attachment for the ovarian ligament. So remember, that's the ligament that essentially provides support for the ovary to kind of help hold it in place. This is also where the blood vessels kind of enter into the ovary itself. And we have two major regions of the ovary. The outer portion of it is referred to as the cortex, and the interior portion is referred to as the medulla. So it's the same thing, the same terminology we used for our kidneys. So what you can see in this image is that we've got lots of little pockets and little cells in the pockets. So this shows you that we've got different stages of development in terms of the ova, so our gametes, our egg cells, and the follicles in which they develop. So looking at the ovary, we've got these ova that are developing in these follicles. So what we're going to do is we're going to follow, just like we did for gametogenesis in males, where we followed the production of these sperm cells. We walked through what the cells looked like and the process that they underwent during meiosis. We're going to do the same thing now for females. And what's kind of cool is the kind of direction that this happens in is similar to what we saw with the males in the seminiferous tubules. So remember, the outermost edge of the tubules, those cells were less developed, right? So they were less defined, or their fates were not yet determined. And then as they went through the phases of meiosis, they headed towards the center of the tubes, towards the lumen. It's the same case in the ovary. The outermost edged cells are ones that are not as far along the developmental pathway as we get towards the center, or the medulla of our ovary. So this is what we're going to be following when we cover meiosis and gametogenesis in females. There are two things that you need to keep track of, and this is what can make things kind of complicated. So we have our oocyte, or our egg cell, the actual gamete that's in the center. That's what has all of the chromosomes that we're going to be passing on. We also have around the oocyte these follicular cells, and the names of the follicular cells actually change. The way we refer to the cells changes based on the number of layers of those cells. And we'll talk about that on the next slide, but I just want to point this out. So the oocytes, or the ova, that are ultimately going to become the egg cells, the ova, develop within this little pocket of cells around them. So that whole thing is called a follicle. So that's the difference. The follicle is the whole pouch or pocket of cells. The oocyte, or the ova within that follicle, is also kind of doing its own thing. So in this image, you can see we've got follicles, or different pouches or pockets, in different stages of development. And the goal here, we're going to walk through with some of my images of what to look for to determine what stage of development the follicles are at and what stage the cells are at. You need to know those things, so if microscopically I zoom onto an actual section on the ovary, you are able to tell me what stages both of these things are in. And you can see that there are representatives of all these different stages in the ovary, and that's because for women, this cycle is around every 28 days. So as the hormone levels change, these cells grow and respond to those hormone changes, but only a few of them do so. So we can always see things kind of in different stages depending on the section of the ovary. Now I'm going to cover the process of oogenesis, so the formation of gametes in females. And this can get quite complicated, so we're going to go step by step, going through a number of sketches that I've created. And I want you to pay attention to two things. One is look at how the follicle changes over time, and two, watch how the actual gamete, the cell that's carrying those chromosomes to be passed on, changes over time. So during development, females are starting out with these undifferentiated stem cells called oogonia. And these oogonia undergo many rounds of mitosis so that we end up with about 2 million of them. These cells then go on to become a little bit more specialized. So we now see a nice ring of squamous-like cells surrounding our gamete. So this happens because right before the female is born, these cells start to undergo meiosis 1, but they stop at prophase 1. So everything starts to get together. We've got all of our chromosomes condensing, crossing over occurs. It stops. Everything halts. So because these cells have started to undergo meiosis 1, we now call them primary oocytes. So you will notice that anytime we've got our actual gamete still with 46 total chromosomes, we call it a primary oocyte. That's our sex cell. With regards to the follicle, because we've got this ring of squamous-like cells, we call this a primordial follicle. And these cells, because we only see one ring of them, are called pregranulosa cells. Okay, so by the time the female is born, she essentially has her complete supply of primary oocytes, right? And they're all arrested in this prophase 1 of meiosis. We call these primordial follicles. And this is all happening, remember, on the outer edge of the ovary, the cortex. Okay. So at this point, all of these primordial follicles hang out, just kind of arrested, hanging out quietly until puberty. And once a female hits puberty and the hormone levels start changing, by this I mean every month we're going to see changes specifically in this hormone called FSH. It's the same hormone that we saw playing a role in spermatogenesis. It's follicle stimulating hormone. It stimulates the follicle to grow. So what does that mean? Our follicle starts to grow. So you can see here that the gamete itself, I've got it showing it becomes a little bit larger. You'll notice this when we look at the slide images. But the follicle itself is starting to grow. So in response to that FSH, things start growing a bit. We still have a primary oocyte because it's got 46 total chromosomes, but because the follicle has grown and we can actually see how the ring of cells in primordial follicles, they start kind of flat squamous like. Now they become this nice ring of cuboid shaped cells. So because we can see this nice differentiation, they're a little bit bigger, we now call this a primary follicle. So this stage does not begin until a female reaches puberty. Okay, next. So because we have levels of this follicle stimulating hormone running through the body, this primary follicle continues to grow. How does it do that? Well, we can see that the pregranulosa cells that were surrounding the oocyte multiply. So they undergo mitosis. So now we see, instead of simple cuboidal epithelium, we now have a double ring of cuboidal cells around our primary oocyte. So when we see, remember this is still primary oocyte because 46 total chromosomes. When we see two rings of these cuboidal cells, we now call the follicle a secondary follicle. Okay, so as long as you have two or more rings around the oocyte, secondary follicle. And because we have two rings of cells, the cells themselves that comprise the follicle are now called granulosa cells. Okay, so some of these primary oocytes are going to continue along the pathway of development. So not all of the cells, so this is something important to keep in mind because we've got these hormone fluctuations every month. So it's just a subset of these follicles that respond to the hormones and actually go along with growth. So a large number of the primordial follicles just simply don't respond and stay where they're at until the next cycles. Okay, so as some of our select secondary follicles are exposed to more levels of follicle stimulating hormone, what we see next, of course, this is getting larger. I'm showing you how we've got more of these granulosa cells surrounding the oocyte. So we call this a late secondary follicle. How do I know it's late? Well, we're starting to see these little spaces that are around the oocyte, and we also see the appearance of two other structures. So we start to see these theca folliculi. So these are cells that form around the oocyte and the zona, sorry about that, pellucida. So the granulosa cells are essentially, we still saw some mitosis because we have a full another ring around them, but there is a liquid that's starting to form in the center of the cells. That's our key that we're dealing with a late secondary follicle, because if you look at the secondary follicle, there's no liquid around it. The other thing that's different is that in the late secondary stage follicle, we see that the oocyte secretes a glycoprotein to form this little membrane called the zona pellucida. That's in grayish. We also see a layer of connective tissue and epithelial cells, these theca folliculi. Okay, so this process just continues. So now we see this liquid is accumulating to a greater extent between the granulosa cells to create this fluid-filled cavity called an antrum. So we now see this fluid-filled space as soon as it's completely solid all the way around our oocyte. We call this a vesicular or antral follicle. We'll also notice that we have many layers of the follicular cells, those granulosa cells, and we refer to it as the corona radiata. Okay, so next, just before ovulation, meiosis 1 finally is completed. So remember, this whole pathway of stuff on the left, I'm showing you every time we're calling our gamete a primary oocyte because it is arrested in prophase 1 of meiosis 1. But right before ovulation, when we've got strong amounts or great amounts of LH, in response to hormone changes, meiosis 1 is finally complete. So, because of that, we end up with two things. We have a polar body. That polar body is essentially a way to dump the extra homologous chromosomes. So the goal of meiosis is to take our 23 sets of homologous chromosomes and separate them so that we're creating cells that only have one set of the genetic information, just one copy of the genome. So to do that, we got to get rid of the extra chromosomes. So that's what a polar body does. And these typically just get reabsorbed in the ovary. But we also create a secondary oocyte. So this is the first time we're finally calling our oocyte a secondary oocyte. And this all happens in response to this big peak of LH, which is also called luteinizing hormone. So we're starting at these peaks in LH. The primary oocyte within our vesicular follicle finishes meiosis 1. And we now have, as you can see in this image, if you look, I now have the label of 23 chromosomes in my oocyte. So it's now called a secondary oocyte. And notice that typically when you're looking at a really large vesicular follicle that's getting ready to undergo ovulation, we refer to that oocyte as a secondary oocyte, right? So in the image above, I'm just showing you that's what we start with, 46. Right before ovulation, it changes from a primary oocyte to the secondary oocyte. And then as soon as we have this huge peak in LH, the secondary oocyte leaves. That's what triggers ovulation. As soon as that secondary oocyte leaves, what we're left with in the ovary, what we can see is a ruptured follicle. So that those multiple rings of granulosa cells still hang around. And that's really important because they're going to turn into a structure that takes over producing different hormones. Okay, So we've got our ruptured follicle. Our secondary oocyte has now left the ovary, so it can begin its journey down the uterine tube. It is not going to complete meiosis II unless it is fertilized. So there's a total difference between female and male gametogenesis, right? Because in males, both rounds of meiosis are completed, but in females, we have lots of pauses and places where things are kind of arrested until another event occurs. Okay. So now this ruptured follicle sticks around in the ovary and starts to become what's called a corpus luteum. So this corpus luteum literally means yellow body. So it's formed from those leftover granulosa cells. And its job, it now takes over the role of secreting estrogen and progesterone. So our corpus luteum gets bigger, it starts to develop, it gets really large as it's taking over and producing all of the progesterone and estrogen. And then finally, if fertilization does not occur, it starts to break back down and degenerate into a corpus albicans. So it switches over to the name of a white body. I kind of drew it orange here, but it's essentially deteriorating. So the two things, I didn't write on here. So this is our corpus luteum, both a developing one and this one's full force over here, producing lots of estrogen and progesterone. But if fertilization doesn't occur, it degenerates, and then we call it a corpus albicans. So it's important because it releases hormones. And if fertilization does occur, this structure sticks around because it's going to take control of releasing hormones that ensure that the lining of the uterus is not lost, because we want to make sure that that fertilized egg then hangs out to fully reach development and become a baby, right? So this corpus luteum structure is really important. Let's take a look at follicular development on a microscopic level. So what we would see looking through the microscope. So just a reminder here that this process of development during oogenesis is going to occur from the outer edge, the cortex, kind of towards the medulla of the ovary, and over a course of 28 days in response to changes in hormone levels. Another reminder, we're going to follow two things. We've got our oocyte, which is the gamete that actually goes through meiosis, and then we have the surrounding circle of cells, which we refer to as our follicle. So let's zoom into our ovary, and here we can see some primordial follicles. So those are all of the gametes that have just a simple kind of ring of pre-granulosa cells around them that are more squamous shape, difficult to see along the outer edge of the cortex. So this is what, as females are born, this is all of the kind of precursors to our gametes. We ultimately start with the primordial follicles. Then once a female has reached puberty, some of these primordial follicles each month will start to grow. They're responding to that FSH, that follicle stimulating hormone. So they're getting a little bit bigger, and those pregranulosa cells start to look more cuboidal in shape, as you can see in this image. So we see one ring of cells. So those cells are pregranulosa cells. Because we just see one ring of cells, it's a primary follicle, and inside we still have a primary oocyte. Once this follicle grows even more in response to FSH levels, we now have a secondary follicle. So you can see that the primary oocyte is getting a little bit bigger. It's still called primary oocyte because it's arrested in meiosis 1, but we now see two rings of cuboidal cells. So when we have two rings, at least, we're dealing now with granulosa cells, and we have a secondary follicle. So here are some images of a follicle that's becoming even, or that are becoming even more developed. So here we have a late secondary follicle. So we're starting to see a little bit of fluid fill in between the granulosa cells, and we have more than just two rings of cuboidal cells. We can also see the zona pellucida and some theca cells around the primary oocyte. So we finally come to the largest looking structures here on this image, and we see lots of fluid-filled space. So that tells us we have the antrum present, so we're dealing with vesicular follicles. So very late on in development. Now, up until this point, the oocytes were primary oocytes. Now remember here, right before ovulation, meiosis 1 is finally completed, so now we have a secondary oocyte. LH has a huge surge, so that's luteinizing hormone, has a huge surge around day 14. So ovulation occurs, that secondary oocyte leaves the follicle, and we're left with a ruptured follicle. So we still see some fluid in there, and the coloration is the same, but the oocyte is gone. That follicle stays in place, and that's really important because it's going to take off over secreting hormones, and we refer to it as the corpus luteum. So look how large this structure is. So we still can see there's fluid on the inside, but it's not as granular. And this structure just sort of looks like it's still the follicle, but it's sort of changing in shape, where it's kind of changing in texture, if you will. It has kind of scalloped edges. So after day 28 and no fertilization has occurred, and we're going to lose the endometrial lining of the uterus. Everything's going to be starting over again. That corpus luteum essentially starts to break down. It kind of shrivels up and is going to ultimately kind of be more absorbed into the ovary. So we can see kind of the scars of past corpus luteum or corpus albicans because it's starting to degenerate. In addition to all these changes that we see going on in the ovary with regards to follicle development and meiosis that's happening with the oocytes, we also have changes that are occurring within the uterus, specifically changes going on with the endometrial linings. And this is because once that secondary oocyte is ovulated, it hangs out arrested in metaphase 2 of meiosis 2 until it's fertilized by a sperm. And this happens in the fallopian tube or the oviduct. Once that happens, that fertilized egg needs a place to grow, receive nourishment, be developed. And that's going to happen within the uterus. So ultimately, all of the hormone changes are preparing the endometrium for implantation of the zygote, and it needs to nourish that developing embryo. So we're going to follow along what happens with our endometrium, and we've got two layers that we're going to be looking at. The functional layers that include glands and vessels support work, and the basal layer itself. So that's kind of what holds everything in place. So in terms of your microscope slides, we have 3 different slides, and your job is going to be to tell me which phase of the menstrual cycle that the uterus is actually in. So there are 3 different slides here that I'm going to show you. So our menstrual cycle begins with menstrual phase. And that's because at the very beginning, between days 0 and 4-ish, we're going to reset everything. So here, we're going to start off with the menstrual phase. The lining, the functional layer of the endometrium is going to be shed because we want to start everything fresh. So because we didn't have fertilization occur, remember that essentially our corpus luteum is going to stop functioning, and so this signals the hormone changes, estrogen and progesterone, LH, FSH, everything is dropped, so menstrual phase begins. So what we can see in this image is our functional layers kind of scattered, and that's just because the cells themselves are being shed. So everything is, all these layers of cells are dead and they can no longer function. So we're going to shed them through the vagina. Okay. Now compare that to after day 5-ish, we're going to start building the endometrial functional layer up. So essentially we're rebuilding, getting everything ready just in case fertilization occurs. So here we can see a nice developed functional layer and our basal layer in darker purple. So this is called our proliferative phase. Notice that our cells look nice and healthy again. We're seeing lots of these little circles that are forming. So these circles are the glands that are going to form and extend throughout the functional layer. But at this point, we see just lots of circles and the glands aren't as well developed. Now we have kind of a barrier here at day 14. When ovulation occurs, we are going to move over into the final phase of the cycle, which is referred to as the secretory phase. And it gets this name because essentially all of these glands that we started to see during proliferative phase are going to become really well developed. You can see how elaborate they are. You've got like whole tunnel passageways with lots of white coloration because these glands are so developed throughout the entire functional layer. And that's because we've got lots of glandular secretion of nutrients. We've got lots of blood supply throughout the functional layer of the uterus. And that's essentially this of the endometrium, and that's preparing it to receive an embryo. I'm going to end with this image that's really similar to page 649 in your lab manual, just to kind of wrap everything up. So this kind of summarizes all of the effects of hormones in the female reproductive system and their effects in both the ovary and the uterus. So that's why this gets so complicated, because there's lots of different things going on. So remember, in the brain, we've got our hypothalamus releasing this GnRH, that's gonadotropin, releasing hormone. So that's the thing that's going to stimulate our pituitary to release our gonadotropins, which are LH and FSH. And we can see how these levels of LH and FSH change over time across this 28-day period. Okay. So in response to those changing levels of hormones, we have first the responses in our ovaries. So that is focusing on our ovarian cycle, because our ovaries are going to change in response to these gonadotropins, those LH and FSH levels. And how that happens is via these structural changes to the follicles, right? So when we're dealing with our ovarian cycle, There are ovarian phases. So before day 14, we call it the follicular phase. And that's because we're dealing with a growing follicle. And after day 14, after ovulation has occurred, we now refer to the last part of the cycle here as the luteal phase, and that's because that's where we finally see the appearance of our corpus luteum, right? So the sorry, to remind myself where I'm at. So these components in the ovary are what are responsible for releasing different hormones, our estrogen and progesterone, which when these hormones are produced, that's what triggers the endometrial changes in our uterus. So that's what's driving our uterine cycle. And when we're dealing with our uterine cycle, we have three different phases that we're looking at. So remember, we're starting, kind of ending and starting with, as this all kind of begins anew with our menstrual phase, moving into our proliferative phase, where we're seeing the development of our glands, and finally secretory phase, where we see a really nice development of our duct and vessel supportive network.